This is Mr. Russell, your AP Biology teacher, and today we're going to continue our discussion on biochemistry. And in particular, we're going to be continuing to look at macromolecules. So today we're going to talk about nucleotides. Monomers, they're often referred to as nucleotides. So polymers are called DNA or RNA. And we know that DNA and RNA are made up of nucleotides, so therefore polymers are made up of monomers, which form our nucleic acids. And these depend on the 5-carbon sugars that's present in each monomer. Well, DNA, if you remember back to your early biology, has deoxyribose for the 5-carbon sugar. RNA has ribose for its 5-carbon sugar. And these are the sources of genes and hereditary information primarily. There are two different types. DNA, this polymer is the master million-dollar blueprint. It is kept safe in the nucleus, and the nucleus acts like a vault to keep the DNA inside of it. RNA is a polymer. It's like a cheap 10-cent copy of the master million-dollar blueprint. It is disposable, recyclable. It makes messenger RNA and other RNA molecules. This is kind of like I have a set of notes, and I will keep that, and I'll keep it in several different forms, and that's going to be in my possession. Kind of like my grandmother's cookbook. So she has a great recipe. Maybe you want that recipe. What I would do is I would keep her cookbook, and I might print you off a copy of it. So it'd be a cheap 10-cent copy that I could give you for making her great potato casserole or something. You would have the copy, I would keep the blueprint, I would have the DNA, you would have the RNA, so to speak. Now, if we look at pyrimidines and purines, we're looking at a couple of different things here. So let's focus up here first of all on these pyrimidines. These pyrimidines are made up of cytosine, thymine, and uracil, or C, T, and U. Think about a big name. Pyrimidines is a very small molecule. It has a 1-carbon ring. Over here you can see here's a pyrimidine, here's a pyrimidine, here's a pyrimidine, here's another one. Well, these others up here have got to be left over. They've got to be purines. Now, when we think about the pyrimidines, how do you remember that cytosine, thymine, and uracil are going to be your pyrimidines? There's a couple of different ways to remember that. You can think about, make a little acronym out of that. I always say you can't cut the pyramid, C-U-N-T, cytosine, uracil, thymine. You cannot cut the pyramid. Others might say something like counting steps takes you up the pyramid. C for counting, takes for thymine, and up for uracil. So counting steps takes you up the pyramid. Now, if we look at these purines down here, if you notice a difference, instead of it being a single carbon ring, it's going to be a double carbon ring. So here's a double carbon. That would be a purine. There's another purine. And if you notice that it is adenine and guanine that are going to be our purines. Now, how do you remember those? How do you remember that A and G are your purines? Well, we remember that you can't cut the pyramid. Well, depending on who your favorite team is, you could say that Auburn, A, adenine, is purely greater, guanine, than Alabama. And again, depending on what year it is, it could be the other way around where Alabama is purely greater than Auburn. So, Auburn, purely greater than Alabama. So now we've got our purines and our pyrimidines. Now, when we look at this, we know that if we're looking over here and we say that this is one strand of DNA, well, we'd have to have another strand over here. When we're looking at those, you're always going to have a purine paired with a pyrimidine. And here we are. We can see that we've got our, let me go back to this. If we look at this, we can see that our pyramid is paired with our purine. So, now these are held together by hydrogen bonds, which is going to be right in here, which is a very weak bond. This is going to help so that the DNA can separate so that RNA can come in here and receive a copy of the instructions later when needed. All right. Here we can see these hydrogen bonds again, just in a different diagram. This maybe helps you see a little bit better. Here we've got our hydrogen bonds, and what that's going to do is it's going to link together our pyrimidines with our purines on this side. And it's just a temporary bond. They can break up, they can get back together. I've said earlier that this is like high school dating sometimes, where it's easy to get back together, easy to break up, depending on what is going to be there to separate the bonds. So hydrogen bonds, easily broken. Now, we also not only have hydrogen bonds, but we also have covalent bonds. So I'm going to back up a little bit. So when we look at these covalent bonds, we're talking about the bonds that are going to be holding these nucleotides together here. There's a covalent bond. There's a covalent bond. So it's going to be these yellow right here, covalent bonds. That's what we're going to be looking at. And in particular, we're going to be looking at something called a phosphodiester bond. So we're going to bounce around here a little bit. These phosphodiester bonds, nucleic acids, they are very strong, covalent. Remember, these are going to be the strongest. And this is the phosphate of one nucleotide joining with a 5-carbon sugar of a previous nucleotide. So, here we are. Now let's back it back up to our normal DNA structure that we've been looking at. So in this case, we're looking at phosphodiester bond here, joining it up to that carbon sugar. And we got that one. And it continues all the way up until we have this big polymer. Here's our polymer made up of little monomers. There's a monomer, there's a monomer, there's one, there's one. This will continue to build throughout. Very strong, very weak. Hydrogen bonds here, covalent bonds here. All right. Now, remember that we said that sequencing is everything, and depending on, we know that this DNA sequencing is what causes, sometimes when there's a missequence, which we'll look at later, or a replacement, when we're looking at things not being in the right place, amino acids not being where they're supposed to be, or being replaced, then we can get serious problems that can be caused, such as when we look at sickle cell like we did in our last set of notes. So you need to know that nucleotide sequence is very important, and we know that DNA also makes more proteins, right? We know that this happens through dehydration reactions. This is just kind of a review for you. We can break those bonds with hydrolysis reactions as well. We know that also it's the 5-carbon sugars that are going to hold these phosphates together, those phosphodiester bonds we were talking about. So we've got open bonds there. It allows one phosphate to hold to the next nucleotide, and it continues to build until we have these phosphodiester bonds, which are going to be very strong polymers, which are going to build up to make our DNA or RNA strands later. Now, we're looking at our DNA double helix. We know that Watson and Crick, these were the first guys back in 1953 to be able to build a model of the DNA helix. They know that it is a blueprint for carrying all of your DNA, carrying all of your hereditary information. We know that it's got two different sides of DNA strands. These two strands are said to be complementary. They fit together perfect. So I'm going to back up again on you. So if we look right here, we know that this this side of the strand over here is going to be complementary to this side. So we say that A and T go together, or G and C go together. Our purines go together with our pyrimidines. And when you can see it here, here is our double helix moving up. So these are complementary, they work together. And without them working together, we end up with problems. So one side has the information to make proteins and enzymes with $1 million blueprints. The other side is a protective cap for the $1 million blueprint. It protects the sequence of nucleotides. So the sides alternate on each strand. So we're looking at this, we're seeing the important blueprint sequence on the protective cap, because you want to be able to, it's kind of like keeping confidential information. If somebody says, hey man, let me tell you this, and please don't tell anybody, you're keeping it safe. Well, you're keeping the information safe just like in the nucleus, and this DNA is going to do that. It's going to protect all that information. Now, if we look at genes and evolution, the more nucleotide sequence, genes that are in common, the more closely related the organisms are. The fewer nucleotide sequence genes that are in common, the more distantly related that they are. So in this case, if you look at our chart here, you're looking at saying, well, frogs way down here are not really closely related to us. The only thing they have really in common maybe is that they've got 4 limbs, there's a circulatory system, some different things like that going on. Well, of these organisms, which one's going to be most closely related? Well, in this case, they were saying that gorillas would be. So you're saying that it is only one gene difference in us, or one nucleotide sequence in us. Talk about a total chain length here of 146 amino acids. So the further you get down this chart, the less likely there is in any type of relationship. Now, so let's continue on. Let's look at the phosphorus cycle. The phosphorus cycle, so the phosphorus is initially a component of rock. As the rock breaks down over time, which is the process of weathering, the phosphorus is released into the soil. Some of that phosphorus dissolves into the water as the rains pass through the soil. The phosphorus makes its way into bodies of water, such as lakes and oceans, and is available for producers like phytoplankton to use to help make organic compounds such as phospholipids and DNA or RNA. Well, plants, we know that they are producers because they can produce their own food. They do that through the process of photosynthesis. Well, plants can also retrieve the phosphorus from the soil and use it to make organic compounds too. So when organisms die or decompose and break down, the bodies, or decomposers, they break down the bodies and return the phosphorus back into the soil so that it can be reused. Okay, this completes our notes for part 3 of biochemistry on macromolecules, and we will pick up where we leave off.